In this lecture, we dive into the world of the amazing membrane, a fluid mosaic that surrounds all of our cells, a flexible yet strong structure. Although some of the information we're covering in this lecture will be review, which is always great since reviewing helps to reinforce what we know, we'll also cover other topics that will give you a deeper level of understanding about membranes. And I'm going to start this lecture with a statement that may be obvious to you, but I have to say this based on my previous experience. All cells, whether they're eukaryotic, archaea, or bacteria, have a cell membrane. Every cell belongs to the cell membrane club. The membrane separates the cell from its surroundings, but membranes are also very prominent within cells. In fact, a large portion of a cell's volume is covered by membranes. In eukaryotes, these internal membranes form organelles and networks, but recall that prokaryotes may also possess internal membranes. Membranes have different roles which are all interconnected. As I just mentioned, the membrane separates the cell from its surroundings and it acts as a permeability barrier. And it can act as a permeability barrier because of its structure, as we'll discuss in more detail later. The membrane also serves to segregate function. If you recall from the beginning of the semester, the fact that eukaryotes have internal organelles that are membrane bound means that they tend to be able to grow a little larger than prokaryotes since they don't need to maintain high concentrations of all solutes throughout the cells, but only within certain organelles. So localizing function makes things more efficient. Membranes are also the site of transport processes. They contain both carriers and channels which help to regulate the solutes that can enter and exit the cell. Since the interior of the membrane is hydrophobic, these carriers and channels are essential in order to allow the movement of certain solutes, like glucose, for example. The membrane is also the site of cell communication or signal transduction. Most receptors are located at the cell membrane. They bind ligands that allow them to relay signals to downstream proteins inside of the cell. Finally, membranes are also important for cell adhesion and communication between cells that are in contact with each other. And this is because they contain transmembrane proteins that help cells to adhere to each other and also connect the cytosol of adjacent cells. Before we discuss the properties of membranes and the function of the components of membranes, we're going to begin by discussing the evidence that led us to our current understanding of cell membranes. Even though membranes were not visualized until the 1950s with the invention of the electron microscope, scientists had been trying to determine the composition of membranes for many years before that. Even as far as the late 1800s, there were scientists who were already studying membrane structure. One of these scientists was Charles Ernst Overton, who conducted experiments using root hair cells. These are plant cells. And using these cells, he found that lipid solubility was positively correlated to membrane permeability. This graph summarizes Overton's finding. On the x-axis, we have the lipid solubility, with the higher numbers representing higher lipid solubility. And on the y-axis, we have the membrane permeability. As you can see, the higher the lipid solubility, the more permeable the membrane is to that particular compound. That result suggests that membranes are composed of lipids because compounds that are soluble in lipids can readily pass through the membrane. A few years later, a scientist named Langmuir determined the most probable orientation of phospholipids based on their amphipathic nature. So he layered phospholipids on water, and what he saw is that they formed a monolayer so that the hydrophobic tails faced away from the water toward the air, while the hydrophilic heads interacted with water. But Langmuir's experiment did not make it clear that the phospholipids in membranes were arranged as a bilayer. However, in the 1920s, two physiologists, Grother and Grendel, conducted experiments similar to Langmuir's, which showed that the membrane was indeed a bilayer. They extracted blood from various organisms and they purified blood cells. The blood cells were ideal for this because they lack organelles, so the phospholipids that were isolated from these blood cells could be concluded to belong to the plasma membrane only. After extracting a known number of red blood cells, Grother and Grendel purify the phospholipids from these cells and they spread them over water. They then measured the surface area that was covered by the phospholipids and compared that to the surface area that was covered by the red blood cells. And they found that the surface area of the phospholipids was twice that of the red blood cells. So that suggests that the membrane was indeed a bilayer. The most thermodynamically favorable arrangement for the phospholipids is then to face toward each other and away from water. Recall that when placed in an aqueous solution, hydrophobic molecules interact with each other, and that phenomenon, if you recall, is known as the hydrophobic effect. Depending on whether a lipid has a single fatty acid tail or two, the way in which the fatty acid tails interact with each other is different. If there's a single tail, the lipids form a mycel, but if there are two tails, a bilayer forms instead, and this is because the two tails cannot be accommodated very well in the mycel structure. The hydrophobic effect is not only responsible for the formation of the lipid bilayer, but also for the sealing of that bilayer. If there is a tear in the phospholipid bilayer, that edge will be exposed to water. Since this is thermodynamically unfavorable, the lipids rearrange spontaneously to eliminate the free edge. The fact that these free edges are thermodynamically unfavorable has another consequence. The only way for a bilayer to avoid having these free edges is by closing in on itself and forming a sealed compartment. So this behavior is essential to the creation of living cells. And it is a direct consequence of the amphiphilic nature of phospholipids. Once the overall structure of the membrane was determined, there were still some aspects of membrane structure that remain unclear. For example, it was observed that a pure phospholipid bilayer had a higher surface tension than a naturally occurring membrane, and the surface tension refers to the ability to resist an applied force. This difference in surface tension between a pure phospholipid bilayer and a cell membrane suggested that there must be other components to cell membranes. It was also observed that a pure phospholipid bilayer was almost completely impermeable to ions and to other solutes like glucose and amino acids. But a living cell was observed to be able to exchange these types of solutes with its environment. So there has to be something to account for that difference in permeability. To account for these differences, Davson and Danieli suggested that another component of membranes were proteins. And they were right about that, but their model was flawed. They proposed that there was a thin layer of proteins above and below the bilayer, sort of like a sandwich. This protein sandwich model was inconsistent with observations of the membrane. The membrane was found to be approximately 6 to 8 nanometers thick, and this is once the electron microscope was invented. So the membrane, you can see it here. This is one membrane. It's a trilaminar structure. So this structure was 6 to 8 nanometers thick, and of those, the actual phospholipid bilayer accounted for approximately 4 to 5 nanometers of that thickness. So there was only 1 to 2 nanometers available to accommodate the proteins. So that meant that these proteins had to be pretty small in order to fit into that space. But membrane proteins were found to be quite large, so they would not be able to fit in that small space of 1 to 2 nanometers. It was also found that membranes are susceptible to digestion by phospholipases, and these are enzymes that cleave the phospholipid heads, so they separate the heads from the tails. When membranes were incubated with these phospholipases, approximately 75% of the lipids were digested, and that would not have been possible if there was a sandwich of proteins above and below the membrane, because the proteins would then protect the phospholipids from digestion. Another piece of evidence that was inconsistent with this model of the membrane structure is that it is quite difficult to purify membrane proteins. It often requires detergents that disrupt the integrity of the membrane. So this suggests that the proteins cannot be solely above and below the membrane because it would be much easier to purify them if that was the case. Finally, the protein sandwich model that was proposed by Dafson and Danieli did not take into account differences between different types of membranes. If there was a layer of proteins above and below a lipid bilayer, then all membranes should have the same protein to lipid ratio. But this is not the case. Membranes from different cell types and from different organelles have different protein to lipid ratios. For example, if we compare the outer and the inner mitochondrial membrane, we see that the protein to lipid ratio of the outer membrane is about 1.2, while that of the inner membrane is approximately 3.5. Based on all this evidence, the currently accepted model of membrane structure is the fluid mosaic model. This model states that there is a fluid lipid bilayer that has a mosaic of proteins. Furthermore, many of these proteins are proteins that are embedded within the phospholipid bilayer. They're known as integral membrane proteins, and many of these are transmembrane proteins, which means that they have a segment that passes through both monolayers of the phospholipid bilayer. Finally, the most recent evidence suggests that membranes are not homogeneous. They actually contain microdomains, which are known as lipid rafts. And these domains have associations of specific lipids and proteins that are often involved in cell-cell communication.